In Chemistry Part 2, we are going to look at biochemistry and its importance with interaction with the human body. Biochemistry is more specific in that it studies chemistry and how it interacts with living matter. So specifically for us, we're going to look at the different chemicals, both organic and inorganic, that are essential for human life. When defining if something is classified as organic versus inorganic, you have to look at the composition of the compound. And the main thing to note is whether it contains carbon. So carbon is defined as, if it contains carbon, it's an organic compound. And if it does not contain carbon, then it is considered inorganic. Water is considered to be the most abundant inorganic compound found in the human body. It has many different properties that you can see in your text and in your PowerPoints that make it very important for the body. Basically, some of the things is that it has a high heat capacity, meaning it can absorb a lot of heat without increasing its temperature. It has a high heat of vaporization, which means that you can heat it quite high compared to other liquid compounds, and it will not evaporate. It has polar solvent properties, meaning it is a substance that is easy to dissolve other things in. Its reactivity, meaning that the water is highly reactive with some things, and with other substances it's not so highly reactive. And also cushioning, meaning that it provides some amount of protection. So some organs such as our brain are surrounded by fluid, which is made up of mostly water, that if we have trauma to our head, the water will absorb some of the force and protect us somewhat. Do you all remember what pH stands for, or what it is? So pH is a measurement of how many hydrogen ions are in something, and specifically it's the concentration. So when you put brackets around this, it means concentration of. And what pH is it's a mathematical algorithm. So you take the log base 10 of the hydrogen concentration, and it gives you a number between 0 and 14. And so this is considered to be the pH. When you look at the pH scale, 7 falls right in the middle, and that's considered to be neutral. Whereas when you move towards the top of the scale, you move towards 14. And so these things are you can actually see in your book, it tells you the exact pH of these substances over here. But this would be like sodium hydroxide. Whereas when you move towards the bottom of the scale, which is moving towards 0, this is where you're going to have your hydrochloric acid. So this is the acidic side, and this is the basic side. There's neutral. Remember that basic you can also refer to as alkaline. And what these numbers really mean is that the concentration is 10 to the minus 7 number. Remember, we're speaking of the number of hydrogen ions. Whereas when you move towards 14, this will be 10 to the 14. And this is 10 to the 0. So each time, so this should be minus 14. Each time you move one step up or one step down, you're increasing or decreasing it by a factor of 10. So to say that there's a substance that has pH 2 versus pH 3 would mean that the pH 2 has 10 times more hydrogen concentration than the pH of 3. So a reminder that organic means contains carbon. There's 2 exceptions to this rule. Carbon dioxide and carbon monoxide are considered to be inorganic substances. When I mention the word carbohydrate, what do you think of? Those of you that are into nutrition and reading your labels of your foods. What are carbohydrates? So these are things that contain sugar. So maybe you think of, pasta, sweets, rice maybe, fruit. These are things that are all coming to mind. Cake. Who likes cake? What about lipids? Okay, lipids. So these are fats. So these are oils. I think of like butter, fried foods. You guys been to the fair lately? Actually, they take butter and they fry it. It's pretty ridiculous. Then we have proteins. So when I think of proteins, I think of meat. So red meat, chicken, pork, maybe beans, nuts. So these things have protein. Then we have nucleic acids. When you think nucleic acids, I don't really think of food. I just think of DNA and RNA. Which, I mean, when we do consume plants and animals, we are consuming DNA and RNA of those animals or plants. Some terminology you need to be familiar with is a monomer versus a polymer. Okay, a monomer would be a simple subunit of one of these organic compounds, whereas a polymer is many. So remember, poly means many, whereas mono is singular. Taking a look at our first class of compounds, which are carbohydrates. I've pulled these pictures from your book to show the monosaccharide versus the disaccharide. So monosaccharide is the simplest form of a carbohydrate. You might recognize this one over here, glucose. Maybe you recognize fructose. Okay, so these are all types of sugars. Remember when you see ose as a suffix on the end of something, it's going to mean sugar. So you see ose, ose, ose. These are all suffixes indicating that the particular compound is a type of sugar. Now, we look at mono meaning singular. Now, it doesn't just mean it's a simple structure, because if you look at the structure of glucose, it is a quite complex structure. It's a 6-ring, so remember hexose means 6, whereas pentose, so pentose over here means 5. So it's talking about the number of carbons found in the structure. Now when you take, say, a glucose molecule and a fructose molecule and you synthesize them and put them together, this is going to give you a disaccharide, meaning 2, so di, meaning 2. This is going to give you sucrose. If anyone knows what sucrose is, it's a very common thing you find in your house. This is just table sugar, so just regular sugar. And you might have heard of maltose, you probably heard of lactose. This is the one found in dairy. So it's the sugar found in dairy products. We also have polysaccharides. So remember again, poly means many. And you see here it's not just one or two, it's a lot of glucose molecules that have been linked together to form a long chain. So it's a long chain, as it says, of monosaccharides. And specifically what we're looking at here is a compound called glycogen. And glycogen is very important for the storage of stores sugar molecules in your muscle tissue. If anyone's ever done races before, if you ever ran a race or a long, like a marathon or a 15K or even swimming or triathlon, you've probably done something called carb loading. So carb loading, which involves eating a lot of sugar in the days leading up to the event, so that the glycogen will be stored in your muscles, so it's readily available for your for whatever the activity is you're doing. And you're also supposed to be relatively inactive for those days, so it's like laying on the couch eating carbs, which, I don't know, for me that's what I do all the time, so I'm like constantly carb loading. And how this works is that when we need to retrieve the sugar, so we in need, it starts to cleave, so it starts to break off these things one by one. So you see here, if you were starting to break these down, you're going to get a lot of glucose molecules out of one molecular compound of glycogen. The next category of organic compounds is lipids. And lipids are different than carbohydrates because they're mostly insoluble in water. They come in a few forms, so the two really important ones, well, they're all important, but the first two, triglycerides and phospholipids, we're going to talk about a lot. Steroids also important, cholesterols fall into this category. And then eicosanoids, number 4, they are kind of a minor, but still very important for some of the physiological processes in the body. The first of these, triglycerides, is important because it has very important functions for the body, such as energy storage, insulation factors, and also protective mechanisms. These triglycerides are neutral fats, which is important to note that these are neutral, so there is no polarity inside of these compounds, and overall they are neutral as well. They contain a glycerol head and 3 fatty acid chains. So when we're looking at the synthesis reaction here, so looking at the final product over here, we have the glycerol head and the 3 fatty acid tails, and this 3-to-1 ratio is important. So 3 fatty acid tails to 1 glycerol head is important, and it gives it the name. So look at the name triglyceride. The 3 obviously then refers to the number of fatty acid chains that are bonded with the glycerol molecule. They come in several forms, so you probably have heard of terms such as saturated and unsaturated fatty acids. So saturated fatty acids typically are solid at room temperature. They come typically from animals, whereas your unsaturated fats typically are liquid at room temperature, and they come from plants mostly. Looking at the unsaturated, they also come in several categories. So you might have heard of trans fatty acids versus omega-3 fatty acids. So typically the trans are going to be associated as bad fats, whereas the omega-3s are going to be good for the body. And there are certain interactions that take place with these two. And in chapter 24, we will go over that more. The next class of lipids are phospholipids. And phospholipids are similar to triglycerides in that they do contain glycerol and fatty acids, but the ratios are different. So a phospholipid has the glycerol as its head, along with phosphorus. So that's different. So phosphorus, and then it has 2 fatty acid tails. So not 3, but 2. So you see some differences between the structures of phospholipid versus triglycerides. And you see down here at the bottom the phosphorus containing head and the glycerol, and then you see the 2 fatty acid tails. When you draw a schematic of it, it's typically drawn like this. So in my own schematic drawing here, you see that it has a head region and it has a tail region. And important to note that the head region is polar. Okay, so if it's polar, is it attracted to water or does it repel water? What do you think? The answer is that it is attracted to water, therefore it is considered to be hydrophilic. Whereas the tail region is nonpolar. It is not attracted to water, so there is therefore said to be hydrophobic. And we will talk more about the phospholipids in chapter 3 because this is what makes up the plasma membrane of all of our cells. So it's called the phospholipid bilayer, and it makes up the plasma membrane of our cells. So the next class of biochemical organic molecules is the protein. Proteins are important. They have many functions in the human body. So as we go through the rest of this textbook, in both AP 1 and 2, we're going to see many different types of protein. We're going to see hemoglobin and keratin and myosin and actin. So there's going to be enzymes. So we see lots of different varieties and different functions of proteins. Make sure you know that proteins have what's called a peptide bond. So it's a special type of bond that holds together its monomers called amino acids. It's a type of covalent bond, but it's more specific. When you see peptide bond, you know that it's for proteins. And proteins have 4 levels of structure, which we will cover. And these amino acids, there are 20 different varieties of them. They all share a similar base structure, but then they have what's called an R group. So the R group is what's different about all 20 of the amino acids and gives each of them their specific structure. When we look at the structure of proteins, it's important to look at how the amino acids interact with each other. So the first structural building of a protein is called a linear or primary protein. So you see primary proteins have just a linear shape, so they're just in a straight line. If you look at your textbook, this is figure 2.18a, so primary structure is listed there. For secondary structure, which is a little more interactive, you see that the secondary structure really shows amino acid interaction, whereas primary is more of just the amino acids laid out in a straight line. So this one shows that there are 2 different types of secondary interactions. Alpha helix, which is more like a spiral, if you guys know what a helix means. And then beta sheet is where you have linear structures that interact with each other. So it would be such as you had 3 linear structures that were lined up as such. And with these structures, it's hydrogen bonding. So I'm going to draw these little dotted lines because this indicates hydrogen bonding that are pulling or attracting different parts of the structures together and holding them together to form these secondary structures. In a tertiary protein structure, we see there are multiple alpha helices and beta sheets interacting all together. So this is a multiple polypeptide chain. It ends up looking like kind of a big glob. So we call this a globular protein. So globular refers to a structure that's tertiary and it has multiple polypeptide, so polypeptide, which means multiple amino acids linked together by a peptide bond, multiple polypeptide chain. And with quaternary structure, you're going to see that there are multiple tertiary, so each one of these structures here would indicate that it's a tertiary structure. So it's the interaction of 2 or more polypeptide chains individually. So you kind of get this larger structure. You can still kind of see the individual tertiary structures, but they're all interacting with each other. So the definition of quaternary is 2 or more polypeptide chains, each with their own tertiary structure. So in this case, I've drawn 3 different ones, 3 different tertiary structures that are all interacting to form what's called a functional protein. Functional protein. So with the last class of organic compound that we're going to cover is nucleic acid. And nucleic acids are considered to be the largest molecules in the body. They're made up of monomers called nucleotides. So these are the smallest building blocks of nucleic acids. And there are 2 major classes of nucleic acids, and we've already mentioned them, so DNA and RNA. DNA and RNA. So we know that DNA, which is what we're looking at in this particular image here, is the building block for life, basically, and it is the blueprint for the synthesis of protein. So when we look at each other and we all see that we have different color hair or skin, This is because our DNA indicates to the molecules and the cells that are building protein in our skin and in our hair the type of pigments to put into it. So it's really, when you look at like the human race as a whole, and it's why people don't really look the same. I know that there are people that resemble each other, especially if you are a brother and sister or brother and brother and have similar family genetics. But the only way you can really look exactly like someone is if you have an identical twin, and it's because the DNA forces us to look different. DNA is arranged in a double helix, so you should see down here that there are two spiral purple structures here, and these purple structures indicate the backbone of DNA. The backbone of DNA is deoxyribose, so it's a sugar, and also a phosphate. So these two structures are important because they create the basis for which the nucleotides can hang on to it. So we have nitrogen bases which make up these nucleotides, nitrogen bases. So you see here the very end of each of the structures that we're looking at down here are a nitrogen base, and they come in four different varieties. They're called adenine, which is designated by A, guanine, which is G, thymine, which is T, and cytosine, which is C. And those of you that have taken biology, you might remember what's called the base pairing rule. So base pair, meaning you always see A and T bound together, and you always see G and C. So they're complements, meaning when you look at this picture over here, you see an A and a T directly across from each other, always. Okay, and you see a G and a C always. Now, it looks like this picture's been distorted somehow, so I would look in the book. You see C and G here, but then it gets to look a little funny down in this area. Look in the book at this figure, and you will see that you always see the A's and the T's across from each other. The figure is figure number 2.21 in your textbook if you want to go over and take a look at that. Now RNA, on the other hand, RNA is a single strand, so it's a single helix, and it also is important for protein synthesis, and it works along with DNA and helps DNA. Remember, it's the R stands for ribonucleic acid, so it's not deoxyribo, it's ribonucleic acid, and it comes in three different varieties: messenger RNA, ribosomal RNA, and transfer RNA. Three varieties. It actually comes in a few more, but these are the ones that we're going to discuss in more detail when we get to chapter 3. Adenosine triphosphate is also worth mentioning. Adenosine triphosphate, aka ATP, is a type of molecule that when bonds are broken, it releases energy. So when we use glucose in a process called cellular respiration, we form ATP from this, and then we can store the ATP to power in the future reactions, whether it's heat or whether it's movement. So there's all kinds of reactions in the body that are going to need ATP for their processes to take place. This concludes our study of chemistry. Thank you for watching. It is, check back chapter 3. The cell will be upcoming next week.